So broadly speaking, we're interested in how mountain pine beetle infestation affects seedling regeneration. And before I go into some of the background on our topic, because this is the first time that we've been that we presented at this session, I just wanted to introduce our research team. So I'm Justine Karst. I'm the lead PI in this project. There are 4 other PIs: Suzanne Simard from UBC, Nadir Erbligan and JC Cahill and Janice Cook, all from UofA, and Nadir and Janice are in the audience today. There are 2 graduate students working on this project, so that's Paul Cigan and Greg Pec, and as Keith mentioned, they'll both be talking after me about their specific components on the project. We have 2 undergraduates, Morgan Randall and Alex Sawinki, and they help us in both the field and lab components of our project. The majority of our project is funded by NSERC, and we have some help also from the Alberta Conservation Association. We're collaborating with the Foothills Research Institute, and we are supported by several companies from the forest and biotech industry. And of course, we've also had some help from the province too. Okay, so as I mentioned, we're interested in how mountain pine beetle infestation affects seedling regeneration. And in particular, we're trying to characterize how site conditions change and understanding the ecological implications of those changing site conditions. And what makes our project maybe a little bit different is that we're focusing on below ground mechanisms. So we're really interested in what's going on in this box, right? So what's going on in the soils. But before I start talking about below ground mechanisms, I just want to go over some of the above ground changes that we expect with mountain pine beetle infestation. So we know that light, nutrients, and neighbors are all important to seedling regeneration. And these 3 factors are likely also to be infected by mountain pine beetle. So I'll just focus on the first 2. So as we all know, so when a tree gets attacked, it fades from being green to red. So the needles, they quickly die and they drop to the ground. And then with that, and eventually also the twigs and branches fall off too. So with that, we expect an increase in light to the understory, and then also possibly sort of a flush of nutrients with this nutrient deposition. And so we see these on the above ground, but we also think that these site conditions will be expressed in the below ground. And so this brings us to the box. And so our overall objective in this project is to look at how below ground mechanisms mediate changes to above ground site conditions and then the implications for seedling regeneration. I forgot one part, sorry. So the other thing that I also wanted to mention is neighbors. And so this is something that we're going to focus on too. And so what I mean by neighbors is, so along with light, nutrients, these types of changes, we also have the death of the trees themselves. And so this will impact the above and below ground community too. So when I talk about the below ground community, what we're really focusing on is ectomycorrhizal fungi. And so that's just what these things are here. And ectomycorrhizal fungi, so if you look in forest soils, it's full of ectomycorrhizal fungi. So hyphae make up a large part of the biomass in these soils, and many of the root tips on trees and seedlings will be colonized by these fungi. And so essentially what these fungi do is they sheath the root tips. So they cover the root tips and they create this interface between, say, tree hosts or seedling hosts and the soil. And we know that ectomycorrhizas, they're really sensitive to soil nutrient levels, and they depend on trees for carbon. So typically this association is viewed as being mutualistic. So there's this reciprocal exchange between trees and fungi for nutrients and carbon. So trees, they photosynthesize, they essentially produce these sugars that they allocate to the fungi, and in return, the fungi, they acquire resources from the soil, especially nutrients, water, and they give that to the tree in exchange for these sugars. We also know that ectomycorrhizas are really important for seedling nutrient and water uptake, and they're critical for seedling survival and establishment. So as I mentioned, Ectomycorrhiza fungi, they're sensitive to nutrient inputs and carbon inputs. And so both of these in this system are governed by overstory trees. So as the system is affected by mountain pine beetle, so the beetle comes in, the trees die, the litter falls to the ground, so you have this nutrient input, the trees die, and there's also a loss of carbon into the soil through Ectomycorrhiza fungi. So these two things are likely to impact the community, but not all species of Ectomycorrhizal fungi will respond in the same way. And so that's why we think it's likely that we could see a shift in the composition of these mycorrhizal communities. And that's really important because previous research has shown that seedlings respond, or their growth and their survival, it can be modified by which species are colonizing their roots. So essentially what we suggest is that through mycorrhizas, there is a connection to, or from what's going on at the stand level to things going on at the seedling level. Okay, so when I talk about ectomycorrhizas, we're really measuring two things. So we're measuring the community or the community composition and the functioning of these communities. So at a root level, so these are just pictures of different kinds of mycorrhizals you'll see in the soil. So at the root level, and if you look at an individual tree, one tree might be colonized by several different species of Ectomycorrhizal fungi. And if you look at the stand level, often you'll find greater than 50 species that you can identify in the soil. And so again, shifts in the carbon and nutrient inputs are likely to change the composition of these communities. And then, as I mentioned, we're really interested in looking at how the functioning of these communities will change as well. And so with the function part of it, we're really focused on understanding the role of mycorrhizal networks. And so just to explain a little bit about what a mycorrhizal network is. So I've drawn here, so here's a lodgepole pine. These are some roots. They look a little bit funky because it's sort of hard to draw roots with PowerPoint, but so there's its roots. And then you have, say, another lodgepole pine and its root system, but this could be a lodgepole pine or it could be a different tree species of the boreal forest. And then essentially, the mycorrhizal fungi connect these root systems. So if you looked at an individual root, you'd see that there's this web of hyphae extending from this root tip. And then this web of hyphae will then connect another root, possibly of another tree. And so same or different tree species can be functionally connected underground through these mycorrhizal networks. And so what research has found that often what you'll see is these larger trees can subsidize the resource needs of smaller trees through this mycorrhizal network. And so seedlings really benefit from this. And so when you think about a living forest, before mountain pine beetle comes in, these large overstory trees are really important to the integrity and functioning of this network. And so what we're interested in is, so what happens when you start to lose these trees through mountain pine beetle attack? What happens to the network? And so a lot of our thinking on this topic has come from information coming out of Suzanne Simard's lab. So she really is pushing and has done a lot of work on looking at mycorrhizal networks. And so, for example, this is a graph that one of her students, Kevin Baylor, put together for a paper. And so this is a picture if you're looking down on a forest stand. And so these green stars, these are individual Douglas fir trees. And I just want you to focus on these lines connecting the trees. So the lines represent mycorrhizal fungi. And you can see that essentially through this network, you have this system of trees. And then these trees, these form hubs. And these hubs basically connect to each other. And the arrow that I have there, so that's one large Douglas fir tree, and it's connected to 47 other trees through this mycorrhizal network. And so what we're really interested in is, so if this is the forest before mountain pine beetle, and then if you were to come and look at the forest after pine beetle, and it's taking out these large hubs that are really connected to the network, what happens to the network? So what happens when you're losing that carbon source? Is the network integrity compromised? Is it degraded? Does the complexity change? And then ultimately, what is the repercussions for seedlings coming into this network? What happens to them? So putting this all together, we're interested in how mountain pine beetle infestation affects the composition and functioning of ectomycorrhizal communities, and then its consequences for seedling regeneration. And so we have four main objectives in our project. So the first objective is to understand the inputs into this box, essentially. So how can we characterize the nutrients coming into this box? And so what happens when you have an increase in letter deposition, and how does that affect the soil chemistry and the soil nutrients? And so this is what Paul Cigan is doing. So his name's not nutrients, it's actually Paul. The second objective is we really want to understand and kind of open up this box, and so look at the communities occurring in this box. And so our main objective here is to characterize these communities and see, do we see a shift in the composition of mycorrhizal communities? And so Greg will tell us some of the preliminary work that he's been doing towards that objective. Our third objective considers the roles of neighbors, and so specifically the death of these overstory trees. And so for seedlings, as I mentioned, most of their neighbors are these overstory trees. And so what happens when these trees die? What happens ultimately to the network of mycorrhizal fungi? Is it compromised? Does it collapse? What's happening there? And then especially the consequences to seedling regeneration. Also, as part of this work, we're really interested in just what's happening to the roots in the soil. So do they decrease in abundance? Does the diversity change? Does the density change? Just these sort of basic questions about root ecology. And so here we're really drawing on the strength of J.C. Cahill's lab to help us understand some of the root ecological questions. And our undergraduates, Morgan and Alex, are going to be tackling those questions this summer. So then our fourth objective is determining the relative importance of light, nutrients, and ectomycorrhizal communities. So parsing out their roles. And Greg and Paul they have an experiment set up looking at that right now, and then Paul will present just a little bit of those results today. And then part of this, in this experiment, we're interested in looking at the seedling survival, growth, and physiology of these seedlings. And so Janice Cook has been really helpful in helping us understand some of the physiological changes that we can expect to happen to seedlings. And then also part of this work, which we're not going to present today, is looking at how shifts in mycorrhizal composition will also affect needle chemistry. And so Nadir has been really helpful in guiding us with that research. So with our objectives, we're addressing our objectives with experiments and surveys in the field and greenhouse. And we're using next generation sequencing technology to characterize our mycorrhizal communities, and Janice has been really helpful in getting our molecular work off the ground. We're using 2 species in all our experiments, so we use lodgepole pine and white spruce. We use lodgepole pine because of its ecological and economic importance, and then lodgepole pine we're focusing on, so because in our sites at least, that is the species that's coming up. So that's the one that we do see in the understory, and we're not really sure of the role of that species yet. So on one hand, we think it might be positive in that the presence of white spruce amidst this dying overstory might be able to maintain mycorrhizal networks or maintain mycorrhizal communities that could benefit other species coming in like pine. Or it could be something more negative in that, say, maybe it pushes the mycorrhizal community to something different, which then prevents other species coming in like pine. So we're not really sure of the role, but that's something that we really want to explore in our work. And I think it actually sort of answers or will help answer some of the questions that came up yesterday about forest recovery. So the majority of our work does take place in the field. And Alberta SRD office was really, really helpful last year in helping us find our field sites. And so I don't know if Anina's in the audience today. Is she here? No, okay. Anina, thank you. I know you're not here. So it's really, really helpful. We were looking for field sites at a time that there was fires popping up all over the province, and she still found time to come help us out and make sure that we were on track with our field site selection. So that was really, really great. For our field sites, we were really intent and really interested in making sure that we could find sites that were protected from cutting for the duration of our study, so about 3 years. And so we ended up focusing our search on sites close to the provincial permanent sampling plots. And as part of our site selection, we had several criteria. So we wanted to make sure that our sites were dominated by lodgepole pine, they were greater than 7 years old, occurred to mid to low elevations, and had a mesic moisture regime. And then of course we wanted these sites to represent sort of a gradient in attack. So in the end, we found 11 sites, and we found them all sort of about 60, 100 kilometers south of Grand Prairie, so all kind of falling in the circle. And you'll notice that, so our sites fall in a region where there's recent beetle activity. So last year, Paul Cigan, he surveyed our sites to see, well, to make sure that they did fall on some sort of gradient. So what you're looking at here is basal area of lodgepole pine. Each of the bars is one of our 11 sites. So the white is the basal area of trees that were not attacked at that site. Green is basal area of trees that were attacked either in 2010 or 2011. Red is basal area of trees that were attacked between 1 and 3 years ago. And then gray is the area of trees that were attacked greater than 3 years ago. So based on this survey, we found that our sites roughly fell out into 3 categories. So non-attacked, green attacked, and red attacked. And so from here, I'm going to let Paul talk to you about what he's finding about nutrient inputs and such with mountain pine beetle infestation.
Good morning, everyone. My name is Paul Cigan. I'm starting my 2nd year in the Department of the Renewable Resources, working on my master's, and want to talk in addition to what Justine said today about this issue of how litter and soils might change across mountain pine beetle attack gradients here. And so where Justine left us off is in this phase of dying trees and their input of litter into the system. And the way of looking at this in the context of how we understand mountain pine beetle decay is that under infestation conditions or epidemic population levels, individual trees fall to attack in this sequence of green, red, and gray across 1 year, 2 year, and 3 years. And so what we wanted to know is where the litter deposition across this time sequence peaks, and what that contribution, what the contribution of that litter fall might be for conditions in the soil. And so that's where we're going to go off now with these 2 questions. Does pine litter chemistry and deposition rate differ by stage of mountain pine beetle attack? And do the soil chemical properties differ by stage of mountain pine beetle attack? To give us that first step in this causal context where we're going with seedling regeneration. And so what I did this summer in 2011 was installed 11 30 by 30 meter plots in different areas near these PSPs. And within those 30/30s, I set up 4 6 by 6 meter sampling plots in which I collected mensuration data and forest composition data. I also did seedling surveys in there. In the stands, I deployed 10 litter traps throughout the stand to capture litter throughout the season. I collected that litter 3 times throughout the growing season. It was a 138-day period. Soil nutrients were measured using soil nutrient probes positioned at every litter trap and extracted at the end of the growing season in August. And we also measured soil moisture at just one time using a theta probe at each of these locations corresponding to the litter traps and averaged them across site. What the sites would have looked like is a situation like this where we have a litter trap from which the litter was collected at 3 different points in the season, brought to the laboratory, dried and weighed to get a dry mass, and soil nutrient probes which were extracted at the end of the season to capture nutrient flux in the soil. What we found is that in terms of the pine litter mass that fell, mountain pine beetle attack basal area was the best predictor, and it positively corresponded to that level of mass, in which 70% of the variation was explained by this metric here. And so we definitely picked up a gradient there, where we see more attack definitely leads to greater input of litter across the stands. Recalling our different stand groups that Justine evoked earlier, we saw that by stand type, i.e. control, green attack, and red attack, the amount of litter did differ. And we saw 85% higher litter fall in these red attack stands relative to the control stands. And so that would have been at kilograms per hectare here, about 2,000 versus about 1,200. It turns out that the pine litter phosphorus load was also different between these stand groups, in which our red attack stands had 36% higher phosphorus concentrations than did the control stands here. So not only more litter, but litter of higher quality, at least in terms of phosphorus. When we combine the concentration and the overall mass, concentration of phosphorus and mass of litter together, what we see is, again, a major difference in the amount of phosphorus input, at least coming in from the litter. Here, where we have over a twofold increase here, or twice as much coming in the red versus the control group here. Although the amount of nitrogen in the litter did not vary between the stand groups, we did see that when we multiplied the concentration of litter and the nitrogen in the litter by its amount coming in, there definitely was a difference in the amount of input in the system there. Soil nitrate concentration did differ, in which the red attack stand was about 29% higher in terms of nitrate than was the control. So we're trending upward for various things here, and we'll see that again with soil phosphate levels. Phosphate levels here were higher in the red attack than the control group. Our soil pH was becoming more alkaline. It was trending upward across the gradient here. So we had about 5.7 there for our control, and about 6 pH for our red attack stands. Soil moisture that we captured through that single measure, which we'll do more of in the next field season, also reflected changes in which the red attack stand was higher by about 30% in soil moisture as a percent volume relative to the control. And so what we can summarize here with the 2012 field results is that there are changes happening across these sites, and in particular, the deposition rate in the red attack group is much higher, of not a huge surprise, but that the quality is different is somewhat of a surprise here, and that it leads to differential input into the system. May that be having a contribution to the soil chemical conditions that we've seen in terms of higher nitrate levels in our red attack group, phosphate and moisture in those groups? Potentially, and I happen to believe it does. So bringing us here, the question becomes, if we do have this changing environment here due to these inputs or less uptake, what might be the consequences for these ectomycorrhizal communities and seedling regeneration, ultimately the resilience of these forests? And so to test this in a more controlled situation and understand what the effects of the mycorrhizae are having from these different types of stands on the growth of seedlings, we've constructed this greenhouse experiment here where we're using white spruce and lodgepole pine, and we're testing the effects of light, litter, and the source of mycorrhizal fungi, i.e. the different stands that it came from, on measures of seedling growth here, carrying out from November 11th to about, November 2011 to about August 12th. Preliminary results from that experiment are showing that the mycorrhizal treatment is having strong effects on the growth of seedlings. So we're optimistic that other changes in the growth might be fleshed out across this experiment. But this is only in the greenhouse right now, and we don't know what's really happening with the relationship between mycorrhizal fungi and the seedlings in the field, and that's where Greg Pec is going to lead us off here.
Hi, good morning. Thank you, Paul. Like Paul said, my name is Greg Pec, and I'm in my second year of my PhD through the Department of Biological Sciences, and I'll be looking at neighbors, particularly ectomycorrhizal fungal communities and their networks, and their role on seedling establishment. So if we take a neighbor level view at local site conditions, seedling regeneration can be determined by a number of factors, including light availability, species shade tolerances, root competition for resources, and as well common mycorrhizal networks. And so if we look here, these lines of colors represent different fungal associations and connections. And so connecting hub trees, or mature hub trees, to seedlings. And so there are a number of benefits to these seedlings being connected to this network. One, through water redistribution, particularly from the hub trees to the seedlings, and two, nutrient transfer. Again, in particular, carbon from these mature hub trees to these seedlings. But as well, other nutrients are transferred and are important, including nitrogen, phosphorus, and potassium. And so the question becomes, what are the limits to mycorrhizal abundance, composition, and mycorrhizal network function along this mountain pine beetle attack gradient? Does the mycorrhizal abundance decrease with greater attack? Does the community composition shift? Does it change with greater attack? And if it does it matter? If it does matter, what then does it do? How does it alter the mycorrhizal network function ultimately for seedling growth, survival, and establishment? And so therefore, looking across varying stages of overstory attack, two objectives I am working on. One, to determine how tree seedling establishment is affected by mycorrhizal networks. And two, does ectomycorrhizal fungal community composition differ among tree seedlings? And in order to do that, if we look back at the 30 by 30 meter stand, there are 10 litter traps within the stand. Around each litter trap, lodgepole pine was seeded within or 3 mesh bag treatments, and white spruce was seeded around or in 3 other mesh bag treatments. And these were all around each litter trap. And the goal of using these mesh bags is to really test the function of the network to seedling performance. And so if we look a little closer at each of these treatments, the first treatment allows root and hyphal penetration. The second treatment only allows hyphal penetration to either the lodgepole or the white spruce seedlings. And the third treatment, there is no root or hyphal penetration. And so a number of seedling measurements will be taken on each of these treatments. For the above ground, that will be germination, survival, stem height, seedling stem C, which is a proxy for plant water use, and shoot biomass. For below ground measures, there will be root length, root biomass, and total number of mycorrhizal root tips. Now, besides the seedling measurements, also interested in characterization of the fungal community composition. And this will be done in 2 ways. One, through visual identification, so morphologically, looking at such characteristics as color, shape, and texture of the actual mycorrhiza. And a subset of those samples will be done through molecular analysis. In particular, the goals here are to identify the Ectomycorrhiza at the species level, and 2, to identify rare fungal species. And again, the goal is to categorize these fungal communities. Do they differ between the attacks or the attack gradient? Do they differ between the white spruce and the lodgepole pine? Again, doesn't matter. And if it does it alter mycorrhizal network function for seedling growth, survival, and establishment? Data should be coming in for the molecular analysis in fall and winter, and hopefully results will be presented at next year's meeting. And so I'd like to send it back to Justine for some concluding remarks.
Hi. So just to wrap up, so in our project we're really interested in looking how mountain pine beetle infestation affects community structure and function of ectomycorrhizal communities, and then the implications for seedling regeneration. So as Paul mentioned, to date we found effects so far in that with mountain pine beetle infestation we see an increase in nutrient inputs, and this is reflected in the soils. And so this year we'll really be pushing to characterize those mycorrhizal communities, see if they also show some sort of shift, and then next year we hope to present these results at the meeting. Finally, I'd like to just also point out that so the context of our work right now is, we're looking at how mountain pine beetle affects these stands and how they change mycorrhizal communities, but you could also generalize to ask, what are the role of below ground processes in forest mortality in general? So mountain pine beetle would just be one agent, one cause of forest mortality, but you could extend this to other systems too. So with that, Paul, Greg, and I would be happy to take questions.